Institutes of Health Research for sponsoring the session and the CHVI Alliance Coordination Office for organizing the session. We're going to have three speakers this morning, and we won't have any questions during their talks, but we'll bring the three speakers back at the end and have an open discussion about any of the material that's presented. We're going to have Dr. Marc Ouellette speaking about the funding landscape in Canada. Then we'll have Jamie Mann talk about some of the issues in developing HIV vaccines and how he's addressed them in previous research. Then we'll have Dr. Gary Kobinger talk about the development of the very exciting development of the Ebola vaccine and how that successful development of a vaccine, the obstacles that were overcome in developing that vaccine can be applied, how they were overcome can be applied to the, we hope, the successful development of an HIV vaccine. So without further ado then, I'll introduce Dr. Marc Ouellette, Director of the Institute for Infection and Immunity and Professor from Laval University.
Thank you, Michael. So it's a pleasure to be here today, and we'll talk about the current status and future prospect for HIV vaccine funding in Canada. So this is a talk that the table at 1 o'clock prepared for me, so thank you very much. So next slide. Yeah, so conflict of interest, none. I will never apply to a vaccine initiative on HIV. So learning objectives, so to prevent and understanding the past and current CIHR investment in HIV vaccine research, share the outcomes of the recent CHR vaccine workshop held in March in Ottawa, and provide thoughts on possible future vaccine programs. So one thing I want to say, re-say, and re-say again, vaccine is a priority for CIHR. Vaccine for HIV is a priority for CIHR. So what about the CIHR HIV AIDS initiative? So a little bit of history. So as you know, there's a large federal initiatives on HIV AIDS, and there's 4 federal partners: the Public Health Agency of Canada, for FAC, Health Canada, through FNIB, and some regulatory aspects, Correctional Services Canada, which actually are involved in our implementation science initiative, and that we are thrilled. So this is the first time that CIHR is doing something with Correctional Services and CIHR. And we also have within Government of Canada what is called the Canadian HIV Vaccine Initiative, CHVI, and this is being coordinated by the Alliance Coordinating Office here based in Winnipeg, and this is many departments within Government of Canada and the Bill and Melinda Gates Foundation. So we received $22.5 million for strategic funding, and there's a little bit more in HIV research that is being funded through the open program, so investigator-driven. So this support has fostered a strong Canadian HIV community, and a testimony of this is the success in the open competition. And this is for partnerships, for community member engagement, and for research gaps and strategic funding opportunity. So these are the pillars, if you want, of the HIV initiative. So we have biomedical clinical, we have health systems and population health, we have community-based research, we have the CTN, and we have CHVI. And really we're trying along the years to have a funding in all of those 5 different pillars, and we are very looking at that very carefully so that we have this historical perspectives in the funding in the different streams. So last year at CAHR in Toronto, there was a lunch where we presented the strat plan that many of you in the room here have participated, and I'll go quite briefly into this, but one is enable discovery research, so new biomedical behavioral systems approach to reduce HIV transmission, the understanding of HIV to slow its progression and mitigate the impact of the health and well-being of people living with HIV, and train and support a strong and diverse community of researchers. And yesterday I was at the New Investigator Forum, where I think this is indeed very vibrant and moving forward. So the first one was enable discovery research. The second strategic direction is mobilize research evidence to strengthen prevention of HIV and other STBBIs in key populations by funding the development, uptake, and evaluation of evidence-informed initiatives, program, and practices, health outcomes for people living with HIV, and strengthen the capacity of researchers and decision makers. And the third direction was to promote leadership and stakeholder engagement and accountability in HIV research. I think this area has really shown the way forward for many of the different initiatives at CIHR, and actually we are often using the example of HIV for stakeholder engagement and accountability. So now a little bit more specifically on vaccines. So this is the level of funding on vaccines that will inform vaccines. So in blue, it's non-vaccine research but related. In dark blue, it's vaccine, and in whatever color between dark blue and light blue, it's the CHVI initiative. And so there's been diverse open call and research initiative grants directly and indirectly support HIV research. Some example is the recent mucosal and cellular immune response to HIV infection that was a partnership with the Gates Foundation. There was both open call and the CHVI initiative that was funded. So behavioral and cultural studies of individuals infected with HIV that can guide the design of HIV vaccine clinical trials and inform vaccine strategies, or multidisciplinary team grants. One that has been very helpful through CHVI has been the Canada Low and Middle Income Country initiative that has funded 5 large teams. So this is the HIV vaccine-related research that is funded by CIHR, and you see that there's roughly around $5 million a year that is invested in HIV vaccine research. So some comes from the priority-driven initiative. So this is the $22.5 million that we're responsible for, and others comes from investigator-initiated research commitments. So 3 years ago or so, or 2 years, well, some years ago, many months, we did consulted with CHI, we did a contract with CIHR Consulting on having an environmental scan of vaccine activity in Canada, and that was very much held by the ACO on, Canadian strengths, CIHR strategy, the funding trends in the field, and what could be prioritized. And this, it's a large document, and I'm only showing one key example of this, and some of the highlights that were related to vaccines, because there were others that are intersecting, like in cure, you could have therapeutic vaccines, but we've tried to focus, or at least to put some emphasis on preventive vaccines. And so some of the areas that were highlighted were novel adjuvants, immunogens, and platforms for active immunizations, immunology of HIV infection, and antibody research. I mean, the field of broad neutralizing monoclonal antibodies is really blooming. Then in cross-cutting teams, there was behavioral science, population studies, and co-infection research, and implementation science and capacity building. And we have an initiative ongoing right now, although it's not related directly to vaccine research. So based on this environmental scan, based on the fact that the vaccine is a priority for CIHR and Government of Canada, and that HIV vaccine is also a priority, we have conducted a workshop on March 18, and the objectives of that workshop was analysis of current investment in vaccine research, for HIV, that I've done an environmental scan, comparative analysis of vaccines and other biomedical prevention strategies, because many are in the work right now, and identify priority areas for future potential investment in HIV vaccine research in Canada. And this is the steering group, and many of the steering group members are present here in the room. And Dr. Jonathan Angel was the intersection, if you want, between the steering committee and the advisory group of CHRAC that he chaired. And also, maybe another point here that I would like to raise, we've bring new investigator also within this steering committee, so to have, you know, a diversity of views of how to move forward. So the workshop outcomes was to advise to CIHR on priority and gap areas for future HIV vaccine research, and to advise to CIHR on possible mechanism for engaging the research community in HIV vaccine research. And I want to say that also at that meeting, IAVI was present, and this is part of our strategy also, is to try to discuss with IAVI on how to move forward on some different aspects of vaccine research in Canada, and to try to link that with some of the international efforts. So the priority areas for future HIV vaccine research in Canada, so this was a consensus of the different members. There were about 50 participants at that meeting, and actually the answer was fantastic, because I think everybody we invited at 2 months' advice have accepted, I think, except one. So novel vaccine strategies, including vector, antigen, adjuvant, and immune modulation, is from discovery to preclinical animal to phase 1 trials. Mucosal immunity, you know, there's strength in Canada, that is a recurring theme, so inflammation, resident cells, increase, decrease target cells, and correlates of protection. I have to admit that this is an area that I've heard for many, many years, but I still think that this is important for vaccine development, and this relates to cohort studies as a means to determine correlates. So what was nice, this was a general consensus. I mean, some individuals indicate, well, that's probably the same list that we would have come like 5 years ago. But I think this was a grassroots type of consensus that has happened, and it does intersect quite well also with some of the strengths in Canada and some of the CHI consulting summary and conclusions that they have reached. So with those priority areas, I mean, the team now is working hard on how to see and to move forward, and there's a number of plans, but are fairly early. But we're confident that we will be able to deliver in a timely fashion for HIV vaccine research. So, I think this is exciting times. I mean, we are aware of some development in the vaccine field that are very exciting for HIV. And I think that in this goal of 90-90-90, vaccine will play a role. I mean, if you want to eradicate the disease, at one point you will need vaccines. And at 1 o'clock here, excluding Eric Arts because he's going to be an applicant, but Michel Peel, Jennifer Ralph, and Elisabeth Paget are, now you understand why I was pointing. They're there, they're working hard to make an interesting funding initiative on HIV vaccine, and please, they have a booth. Now I remember that word, which I was not last night, and ask them questions, and we're hoping that you will all apply and that the best science will be funded. So now it's getting yellow, so I'll stop and I'll let others talk, so thank you.
Thank you, Marc, for talking about the funding landscape and the future directions. Marc will be back after the session to answer people's questions that are raised. So our next speaker is someone who has worked on an HIV vaccine. developing the vaccine and optimizing delivery strategies. He started the work in the United Kingdom and also did some at Case Western Reserve University in Cleveland. He's now assistant professor at Western University in London, and welcome to the stage. Jamie Mann will be the next speaker.
Okay, thank you very much. I'd like to say thank you to the organizers for giving me the opportunity to speak to you here today. So today I'm going to talk to you about HIV vaccine delivery. Specifically, the work that I'm showing you was done as a preclinical assessment to a human clinical trial that was funded in the UK. I have no conflicts of interest to show you. And okay, so the learning objectives, I kept that very, very broad because I really didn't know actually how to write learning objectives. So basically, it was basically how to understand and elicit T cell and B cell responses through DNA vaccination. So the work I'm going to show you today is based on a DNA prime protein boost protocol that we optimized. And this is the regimen that we're hopefully going to use in humans. So the idea was, how do we generate a very, very strong, robust T cell and B cell response through DNA vaccination with protein boosting, and how and whether or not we can actually generate protective humoral immune responses, not just in the systemic compartment, but maybe also in mucosal tissues. And one of the things I really wanted to emphasize in this presentation is whether or not alternative routes of vaccination or alternative kinds of vaccination should be considered in going forward with an HIV vaccine. So the question is, why a DNA prime and a protein boost vaccine? Well, for the most part, protein administration or antigen administration is very good at generating strong B cell responses, but it's not very good at generating a T cell response. DNA vaccines, on the other hand, are very good at generating T cell responses, but are very moderate in terms of being able to generate a B cell response. And work has been done that's shown, and I think it's very convincing, that DNA prime protein boost vaccination can actually selectively amplify your B and your T cell repertoire against conformationally dependent epitopes, thereby enhancing the potential for a neutralizing antibody response for HIV. So when we deliver a DNA vaccine, we most commonly deliver a DNA vaccine by the intramuscular route. And that's because myocytes or muscle cells are very easily transfected, and they can actually produce large amounts of vaccine antigen to stimulate an immune response. However, one of the theoretical drawbacks to intramuscular vaccination, and why we believe that DNA vaccines have not really translated well from animal models to humans, is the fact that there is the number of antigen-presenting cells that are present in muscle tissues is low in comparison to other tissues. For example, intradermal vaccines are known to be very, very strong stimulators of immune responses and have a high number of antigen-presenting cells. Unfortunately, intradermal vaccines are not, intradermal vaccines are not very good for DNA vaccines because epithelial cells are not as easily transfected. So our overarching hypothesis for this was, if we were to combine an intradermal and an intramuscular vaccine, can we get the best of both worlds? And just because we wanted to actually generate high T and B cell responses through DNA vaccine, we also threw in electroporation, which is actually a mechanism where we apply a transient electrical field across the site of a vaccine, where we can maybe open up pores in cells and allow DNA to enter into the cells more efficiently. The antigen I'm going to use in these studies is a trimeric GP140, CN54 GP140. And the reason why we're using this trimeric GP140 immunogen is because a number of studies have shown that this protein is a trimer. It's more conformationally accurate than the recombinant GP120s that have been used. And antibodies which are conformationally dependent, that are neutralizing or non-neutralizing, can bind to this antigen. So this antigen does display some degree of conformational similarity to what a native envelope spike might display, although we do know that this antigen is not perfect. And this work was done by showing that the GP140 can bind to these antibodies was done by Dan Stee and by other researchers. So in the first instance, because we're doing a DNA prime protein boost strategy, I'm just going to talk you through the priming stage of this strategy and how we optimize the priming stage. What we can see here is that I'm showing you on the, this side here is the serological response, and here we have the T-cell response. And what we were just doing was just checking that our plasmid DNA, which encodes the CM54GP140 protein, is actually immunogenic, and that it can generate an antibody response if we deliver it by the intradermal or the intramuscular route. And what we're also checking in this is that whether or not electroporation can enhance the humoral or T-cell response. And I think it's fairly clear to say that when we deliver our DNA by the intramuscular route, we can generate a strong antibody response. And that response can be boosted by electroporation. However, if we deliver the DNA by the intradermal route, it doesn't generate a significant antibody response. But we can actually generate a significant response if we do apply electroporation. In terms of the T-cell response, it's very clear that electroporation can augment the T-cell response. It may not be significant, but it does actually increase it. But in certain cases, it does actually significantly increase the response. This is ELISpot data. How we did it was we used 15 MERS overlapping by 11 that spans the full proteome of the GP140, and we just split the peptide pools into peptide pool 1 and 2, depending on which side of the protein the peptide was on. So, going back to the main goal of this study was to look at concurrent or combination delivery of vaccine. So what we did was we compared intradermal vaccination with intramuscular vaccination with intradermal plus intramuscular vaccination, where we split the dose of the vaccine over 2 sites. And what you can see here is the serological response, and here's also serological response, and here we have the T-cell response. When we split the vaccine over 2 sites, we get a significant antibody response, but it was only equivalent to the intramuscular alone group. And both intramuscular and the concurrent IDIM vaccine regimen was significantly different compared to the intradermal group. So it didn't look like it was actually, by using a concurrent regimen, it didn't look like it was actually doing anything in terms of augmenting humoral immunity. Certainly when we looked at the avidity of the antibody response, now we look at avidity because it's an indicator of how tightly the antibody binds to the antigen, which is it kind of indicates whether or not affinity maturation is actually occurring. We looked at the avidity of the antibody response, and we again didn't see very much difference between the intramuscular alone group and the concurrent IDIM vaccination regimen. But certainly when we looked at the T-cell response, we were able to see differences. And I'm sorry that the label is a bit wrong here, but when we look at ID by itself and IM by itself, they look fairly equivalent in terms of T-cell responses. But when we do ID and IM together, they're significantly different compared to the individual vaccination regimens. So that gave us hope that the IDIM protocol might actually be doing something. So we further evaluated this by looking at different iterations of ID and IM with or without electroporation at those sites. And what I'm showing you here is the serological response in these 3 figures and the T-cell response in these 2 figures. In the top figure, we're seeing that despite the different iterations of concurrent vaccination, the antibody response was fairly the same. The kinetics were the same, and you couldn't really tease it out. However, when we looked for the antibody response in the vagina, or in the vaginal vault, and we looked for antigen-specific IgG, we can see that 2 of the vaccination iterations were actually able to generate significant levels of antigen-specific IgG in the vagina. And that was the ID with electroporation plus intramuscular vaccine, and the ID electroporation plus intramuscular with electroporation regimen. When we looked at the avidity of the antibodies in serum, what we can start to see is we start to tease out little differences that are occurring in the groups. Despite the serological response having the same titers as one another, qualitatively the avidity data shows that they're very, very different. And the 2 responses that that generated the antibody in the vagina were the 2 responses that had the highest avidity had the highest avidity in serum. When we looked at the T-cell responses, again, it was the IDEP plus IM and the IDEP plus IMEP groups that generated the highest T-cell responses in these animals. So just to conclude in that little short bit of data there, I just want to say that electroporation does enhance the antibody and the cellular responses that we were seeing, and that the 2 groups that are shown here were the best combinations to generate humoral and cellular responses and generate humoral responses in the vagina. The IDEP and IMEP group, though, generated the highest avidity antibody responses, again indicative of increased affinity maturation of B cells. So we decided to take that last group forward as our DNA priming strategy and combine it with the protein boost. But the question was, how do we deliver the protein boost? Which route do we deliver it by? So we decided to look at various different routes, whether it was the subcutaneous route, the intranasal route, the transcutaneous route, and the intramuscular route. And what I'm showing you here is data from the serological IgG or the serological IgA responses. I think it's very clear here that the subcutaneous antigen boost or the intranasal antigen boost and the intramuscular antigen boost did generate significant antibody responses, and by the end of the study, the titers were fairly equivalent. The transcutaneous delivery of antigen didn't really stimulate a robust IgG response. Really, the only delivery route, protein delivery route, that actually generated significant IgA responses in serum was the intranasal route. But bear in mind that this is mice, and we would expect that to occur in mice. In terms of the T-cell responses in these animals, it was much the same. Whenever we compared any of the DNA and protein boost regimens with each other, there was no real difference in terms of their T-cell reactivity to the peptides. Obviously, there was significant difference between the DNA prime protein boost and, let's say, DNA alone or protein alone. But there was no real difference between any of the groups in terms of DNA priming protein boosting, suggesting that the protein boosting didn't really have a big impact on the T-cell response. When we look at the mucosal responses in the vaginal vault of these mice, what we can see is that all the mice generated an antibody response against the CM54GP140 protein if it was delivered as a DNA prime protein boost, compared to just DNA alone or protein alone. And again, it was only the intranasal group that actually generated significant IgA response in the vagina. But despite having very similar antibody titers and T-cell responses, this is a graph showing, again, the avidity of the antibodies that you generate with the different regimens. On one side, we have the DNA prime and the protein boost. And on the other side, you have just the protein alone. And those little pie charts are the animals in the group and which animals were either had low, medium, or high avidity antibody titers. And you can see here very clearly that the DNA prime subcutaneous boost protocol generated the highest avidity antibody responses with the most animals that had high avidity antibody titers. In other words, the antibody just wouldn't wash off with even with very, very stringent denaturing conditions. So this was the protocol we decided to take forward for the final the final experiment I want to show you, which was done in rabbits. The reason why we did this work in rabbits is because rabbits are actually a very good model for looking at, let's say, functionality of your antibody if you're looking for, let's say, neutralizing activity. So we wanted to look at our vaccination regimen and see if we could actually develop any sort of neutralizing activity. And so we took the DNA prime subcutaneous boost and we compared it to DNA and subcutaneous protein. And what we can see here is that the DNA prime subcutaneous boost protocol generated significant levels of antibody against the GP140 protein, but that was about 2 logs higher than DNA alone or subcutaneous protein alone. Again, what we can see on the graph immediately next to it is the avidity of that antibody response was actually increased when we used the DNA prime protein boost compared to DNA or just protein alone. And significantly, what we saw when we partnered with Mike Seaman and Harvard University to test the neutralizing activity of this serum, we could actually see that the DNA prime protein boost protocol generated neutralizing activity against a host of tier 1 pseudoviruses, whether or not they were clade C, B, or A, demonstrating some sort of cross-clade neutralizing activity. So to conclude, what I'm showing you here is that the concurrent DNA and protein boost vaccination elicited higher IgG antibody responses. The DNA prime protein boost regimen increased antibody avidity, suggestive of increased B-cell affinity maturation, and that the serum of vaccinated rabbits had neutralizing activity against a range of tier 1 pseudoviruses ranging from clades A through to C. So the work was done as a prequel to a human clinical trial. And this work, I think, is actually quite promising because what it does show us is that the roots of immunization is very, very important on how it impacts the immune response. And I think that is something that we really need to take into consideration when we actually take this work forward. This work, I do believe, we are now trying to take forward to the macaque, and so we can actually really look in depth at the immune responses that we're actually generating with this DNA prime protein boost protocol. With that, I'd like to say thank you to everyone that was involved in this study, everyone at Imperial College, the Shattuck group, Simon Jeffs, who helped us with the CM54GP140 protein, people at St. George's in London, INSERM, Dr. Bazine Combadier, Roger Tattoo at UKHVC, Polymoon Dietmar Katzinger, who gave us the soluble protein, Fit Biotech, who gave us the plasmid DNA, and Particle Sciences for their intellectual input, and obviously all to our funders, the Wellcome Trust, Bill and Melinda Gates Foundation, and QTVAC. Thank you very much.
Thanks very much, Jamie. I now have the great pleasure of introducing Gary Kobinger to speak. Gary, I guess, is in the transition of, well, he has been the head of special pathogens at the National Microbiology Lab here in Winnipeg, and either has or will soon be moving to Laval University to become the director of research in infectious diseases. I think that's the affiliation you have on the program, so I'll include that, and it's great. So you'll hear about Gary's contribution to the development of the Ebola vaccine, which is really quite remarkable, a real success story, and he's in pretty high demand now to speak in all different fora, and we have great that he's been able to take the time to speak to us today, and I've also just been informed that Gary's been recognized with the Order of Manitoba. So in case you didn't know, I think you did know. It's not the official announcement, but that just in recognition of the work that he's done, and I think this should be a great presentation. So thanks, Gary.
Thank you so much for for having me here, and lots of stuff to show, so I want to get back to it, but I'm here until June 30th, so people that are in Winnipeg, please don't hesitate to come see me. And moving in July to Quebec. In the meantime, I want to, with that 15 minutes, I want to really try to see how the adventure with the Ebola vaccine can potentially have some suggestion and things to consider for the HIV field. You know, the HIV field is such a solid field, a leading field in the entire world of virology, all the best immunology, the best molecular biology, the best everything, because a lot of resources were put into HIV. And so it's a bit humbling to come up with, from a small field like flaviviruses and try to highlight things that may be of value. And please take all of this with a grain of salt, but maybe they're a good point to reflect on. So most of the vaccine for Ebola, when I say most, is really the large majority, are targeting the envelope glycoprotein. So what you see in red on the left is a computer image of Ebola, quite well done. Actually, was done by a Russian scientist. And on the right is an infected cells. And why is the GP being targeted? It's really from testing all the proteins. So it's really a systematic approach of doing vaccination and challenge studies. I think one thing that influenced, which is also present for HIV, that influenced the Ebola vaccine development is the field experience and going out in the field and really see the reality and what could be realistic as a vaccine. And what you see now actually in phase 3 clinical trials are 2 vaccines that actually are not fully adapted to those conditions, as a matter of fact, because they need to be stored at -80 for now. You know, I think -20 may be likely to be fine, but and it's to say that it's not there yet, the perfect product, and there's still work to do. But these are some considerations that are important. Another one that came up for Ebola is that the fact that transmission between humans is relatively well known, but the emergence of Ebola in the human population, that is a big question mark, and we don't know exactly what's happening in the animal, what caused the virus to emerge completely randomly like this, sometimes 3 times in a year, and sometimes there's nothing for 5 years. And so what this tells you, I think it's that targeting the human population for vaccination is definitely something that may have an impact, instead of going to chase something that we don't know what's happening. But of course, if we could block this emergence into humans, it would be of value, but this is not there yet. I took these slides from Mainz, and this is just to highlight the fact that the VSV vaccine was really an initial idea of Heinz to use a vector VSV pseudotype, and I'm going to show a slide on this, with Ebola, that has been used for years before he started this vaccine work, but he's the one that thought about using it as a vaccine. And after that, a lot of people contributed. Stephen Jones, Uta, Judy Adimonti. So it's, I mean, the list, Tom Geisberg that does most of the non-human primate studies, the list is so long. It tells you one very important thing. It was a multi expertise approach with a lot of players, not only in Winnipeg, but also outside. And again, I think this is something that was important in the development. So what was started at the time is that actually Yoshi Karaoke and others like Victor had built a VSV that we say pseudotype with the envelope glycoprotein of Ebola, the main antigen for vaccination. And so this vector basically was replication competent. So VSV is an animal pathogen. It's not associated with severe and basically any symptoms in humans, but humans, farmers that have been exposed to it do develop antibodies. So the safety question was a big question mark. But this being tested as a vaccine was immediately a very strong candidate and was used for years and years as a gold standard, basically, the vaccine to beat in terms of protection. And this slide is showing a little bit the progression from the first paper in 1997 by Yoshi's group, Madison, that has used this vector more to study the glycoprotein. Heinz came up with the vaccine approach and published this in 2004 with the work being done in Winnipeg. And then Tom Geisberg did a lot of work at USAMRIID and now at UTMB and so on. And what's interesting is at the time, when I was here, we were moving to platform. I was more on the adenovirus, and it was the simian adenovirus we were calling it, from chimpanzee. And interestingly enough, I think we got very, very lucky because both vaccine ended up, the two vaccine, in phase 3 clinical trials in West Africa. And this slide, I think, is important, and it shows you all the vaccine. Ebola is not a big field like HIV, keep this in mind, and I know you know this. Look at all the vaccines, it's not important the name, but these are all the vaccines that went all the way to non-human primate studies. This is years before you can go to non-human primate studies in level 4, and each experiment costs about $200,000. So there was a lot of work done, and so a lot of vaccine were considered and studied. So it's not the all the eggs in one basket approach that solved this problem of Ebola, which was not such a small challenge. And like at any other field, we got to the challenge of, yeah, we have all these great vaccines that are working on human primates. How do we really translate that into a vaccine in the clinic? And this rodent says to the human, well, I think it'll be years before you'll have a chance to be protected. And of course, you know, the big outbreak did help, but I'm going to get back to this quickly. But the other thing that we had, but we didn't have for years, but after 10 years of work on vaccine against Ebola, we finally identified, it was a very controversial field, identified with a very good level of confidence, statistically speaking, the correlates of protection, at least one of the correlates of protection. Not what is necessarily providing protection, but at least we could look at antibody specifically directed against the glycoprotein, the envelope glycoprotein, and predict survival 99.98% of the time in non-human primate. So this was a very important finding, but let me remind you, 10 years of vaccine development went without this. So I'll get back to the conclusion of this, but you know, as correlates of protection are important, they're not essential to make very important progress. This is the list of vaccines that went to clinical trials. It's also a long list, if you look at it, and I think, again, the HIV field has evaluated a lot in different clinical trials and keeps moving ahead. So what made also another success of the VSV, you know, those two vaccines, the Simian ad and the VSV, went to phase 3 clinical trials. Only the VSV has met with initial data showing efficacy, and one big reason is the design of the study. It's not necessarily that it's the only one that works, it's that they are the only one that came up with an old concept that was used for smallpox, the ring vaccination. And because of the cases were already going down, this is the design that allowed them to reach the conclusion that yes, there is definitely, mathematically speaking, benefit associated to vaccination, being that if you're receiving the vaccine after 10 days, nobody got infected. And so, again, something to think about, you know, when also designing vaccine. And of course, it came to that landmark paper that, again, I said, showed after a preliminary report, efficacy of the vaccine. There's still work to do, you know. There was adverse effect. They are not severe in most cases, but there was inflammation of the joint that was reported in Europe, much less in Africa. So, you know, we don't have the perfect vector vaccine yet, but it was such a tremendous step forward that, you know, other strategies, other vaccine can try to see now how much they can improve this. So what is another aspect? Why is the VSV vaccine that was thought for so long to be will face safety problem? Because VSV causes neurovirulence if you inject it intracranially in animals. This was found out in other vaccine trials in HIV, using VSV for HIV trials, for example. And of course, intracranial injection is not a normal way to vaccinate people. That being said, it is part of the toxicology studies that are being conducted in vaccine development. And the one thing that did help a lot for VSV is that there was this compassionate use in 2009. A German worker in the lab pricked herself with a needle. It clearly was drawing blood, so it did break the skin. The question was the needle contaminated? Anyway, Winnipeg sent were very fast, 36 hours. That lab worker was offered the vaccine and decided to take it. So there was no serious adverse effect with this N of 1, but it's still, there was some level of understanding from the community that it was something to consider, a vaccine to consider, which did happen during the big outbreak. Another thing that did happen that I'm not sure is applicable to HIV, but the compassionate use of ZMapp, the treatment, into 2 Americans very early in the outbreak, so in July 1st, as a matter of fact, of 2014, prompted discussion, ethical discussion at WHO within 2 weeks. And this was a huge push for all the vaccine and all the treatment to go and advance. And again, this is a map during the tough moment of the outbreak, and you know, each location is like a war zone when you look at it. There was a lot of transmission. It was really a tough situation, and prompted, you know, again, advancement of those vaccines. But when you think about it, there's a much higher cost from HIV in terms of lives. So to not also advance things has a real cost. So this has to be put in the balance as well. So I think that what I'm trying to tell you also with this is that, and it's part of my conclusion, is that, you know, if there is to be an HIV vaccine sooner than later, it means that a lot of vaccine needs to be tested, in my view, empirically. So I'm going to give you an example, a project I've been working with Yao, and I forgot to ask if he was going to come this morning, but the first time we present those data ever. And it's a really, you know, project that we, daring project, I think I'll call it, that we have been working on for years now, but really to use as a how can I say that? Prototype experiment. And what we did is, when you look at HIV and Ebola, they both don't have licensed vaccine. HIV has the advantage of having very good therapies in place, and Ebola is advancing, but the big difference is the public health impact of HIV is much bigger. So Emisa, who is here in the room, has been working on this project for a master, and she tried to see if we can mix HIV and Ebola and come up with actually a superhero vaccine with this. And the idea of this is that the problem with, one of the many problems with HIV is it targets CD4 cells, as we all know, and this knocks down our capacity to really respond to efficiently to the virus. So Ebola doesn't do that, and what this approach is doing is it's retargeting HIV to other cell type than CD4. So CD4 are not being touched at all. The immune response is being kept intact, and this virus, which is replication competent, is basically using the Ebola envelope to retarget HIV particles. And the beauty of this is that the virus replicates, expose a multitude of antigens. So I call it an evolving vaccine, basically, where a lot of antigen of HIV gag, for example, are presented. The immune response can control it and shut it down, but is being exposed to a lot of antigen at the same time. If there's any escape mutant, it also can be taken care of by the immune response. And so those vectors do express the Ebola glycoprotein. They do form virus particles. They can, on top, they can express reporter genes. And at the bottom, interestingly enough, in the model world, they don't replicate, so normal mice. They do generate an immune response that led to partial protection against Ebola virus with a single dose. So the bottom line is in summary, that VSV Ebola did not cause the next zombie apocalypse, and we're happy about that. And I see the next zombie apocalypse because people that believe in zombie apocalypse, they're convinced that there was one already, or many. We think HIV Ebola will not cause the next zombie apocalypse. We were glad to see our biosafety officer agreeing with us. I think the bottom line of this is that we need novel vaccines and approach, and that they need to be screened out, and many of them. And correlates of protections are very important, but not essentials to vaccine development. Vaccines are essentials to vaccine development. You can see the rest. You know, I think that HIV vaccine has such many huge challenges. Otherwise, that would be a vaccine already. But I think they are actually very interesting challenges. And I hope that the bottom line is that it's very important, I think, that Canada contributes to those efforts because they are economic drivers. They are important to bring keep affordable healthcare. And they are very important that even if we get rid of all the HIV cases in Canada with drugs, it won't stop coming in, or people are traveling, 4 million of Canadians are traveling every year. It won't stop coming until we have a global approach to HIV infection. That I know you all know that, so I'm not teaching anybody here anything, but wanted to mention it. So thank you so much. I hope that Emilissa is in the room. She's the one with the blue shirt. There's many blue. I cannot point, but she's right next to me, and she's in this room. If you have questions about the project, put a picture of Yao. Yao also have been working very intensively on this project. And that's it. Thank you so much for your attention.
Staying up here. So thanks very much, Gary. If Marc and Jamie would like to come up, we have an opportunity for questions for all three speakers from the floor. Obviously, very diverse talks, but also obviously very common theme, and I'm sure they'll be happy to take questions. Do you want to stand? It's up to you guys. Do you want to move? Not behind anywhere but behind So, any questions from the floor? Find that a little hard to believe. Robert, would you like to ask anything? I'm sure you would. Don't be shy. Don't be shy, Robert.
Since I'm forced to ask a question, Gary, that was a beautiful talk, and I wonder if you could elaborate a little bit, I think, on your suggestion about how to compare a number of sort of simultaneously interesting candidates in clinical trials rather than what I think is sometimes characteristic of the HIV field, where we do one and one and one sort of in isolation from each other, and comparing responses has been a very difficult challenge for HIV vaccines, but yours was sort of a step forward about that issue.
Yeah, that's a very good point. Oops, sorry again. That's a very good point. I think that was an advantage of having the Ebola vaccine being done at the same time, kind of side by side. But keep in mind that they were using different design, very different design. And again, the chimp ad did not reach the conclusion of efficacy, but it doesn't mean it doesn't work. It's mainly due to the design. They had a large, completely randomized approach, not the ring vaccination approach. So at the end, it was very hard to compare. I think the advantage was the timing, maybe. But as I understand, and I'm not following the HIV field as closely as maybe I should, but in the U.S., the NIH has evaluated in the clinic all the different, several, not probably all, but several vaccine approach, including DNA and AD and so on and so forth, and in an independent manner, so to speak, and came up with the conclusion that one of them was probably, had good chances to make it. So actually, I could reverse the question to you. So I don't know what you think about this approach. I think it's, to me, a reasonable thing to do, actually, from maybe a national perspective or investigating, getting together and actually, getting those things in the clinic in phase 1, where you can get very good immunogenicity data, and same thing in non-human primates, and just do it side by side in an independent manner. So an independent site or something like this, I think, is worth years and years and will save resources, and is, to see something next to one another. Any investigator will admit that if their vaccine needs improvement, well, they need to know that first, and they will see it and will admit it and say, well, let's get back into work, or just concentrate on this better one and make it better with the knowledge we have. That's my opinion on this.
I have a question for you. So, you've shown that you use a HIV backbone for Ebola vaccine. Like the Ebola vaccine was on a VSV-based backbone. So what are the progress on making a HIV vaccine through the VSV backbone?
Yeah, so Mark Feinberg presented some very cool data on this, like where, initially this was an older idea, so to speak, and it was tested, it was not great. At the end, it was kind of put on ice, and that was VSV expressing HIV, but also expressing the G of VSV, which is also a bit what brings the neuro targeting. Actually, what he showed by using only HIV envelope on VSV, much better result in non-human primates, including in a challenge model. I think, honestly, I didn't want to show what we're doing with VSV, but I think there is great progress to be made also with VSV against HIV, depending what approach you're using, yeah. And many other, I think that the DNA actually was the one that was selected, as far as I understand, in the US as the most promising platform for now to move forward, right? So I think it's very exciting to see a DNA vaccine that initially, nobody really believed into, and now it's being the leader. So it is to tell you how you can improve things.
Any other questions? I have a question for Jamie. So much of the work, the Ebola vaccine, the preclinical work was non-human primate work. There's a lot of enthusiasm for humanized mice, development of humanized mice as models for HIV. You presented some rabbit data. We don't see a lot of rabbit data presented in the HIV vaccine field, at least I don't. Are we missing a better model that we should be exploiting to a greater degree, or what are your thoughts?
Well, the vaccine, the rabbit model is it is used. It is used for looking at neutralizing antibody activity, and it's got to do with, from what I understand, something to do with the CDR3 length, which actually helps in terms of evaluating neutralizing activity. It is something that we should actually be using, but obviously the macaque is the model that we really need to have in order to actually test whether something's actually going to be effective. So.
Yeah. Anyone else? Feel free.
So this question's probably for you, Jamie, maybe for you, Gary, as well. There's a justifiable focus on broadly neutralizing antibodies against HIV, but most people in the room probably know a lot of the issues with trying to induce broadly neutralizing antibodies against HIV. Do you think there's a route forward for any kind of conventional vaccine antigen and delivery system that will work to induce broadly neutralizing antibodies against HIV?
You go ahead.
That is exceptionally difficult to answer. I really don't really know. I mean, possibly. That's the best answer I could give you. Sorry.
I'm gonna challenge this concept of the importance of generating broadly neutralizing antibody. Again, I think it's actually risky to conclude that this is important necessarily and that this will resolve everything, and by focusing an approach that will generate broadly, and I'll take many examples. Ebola, by the way, neutralizing antibody do not correlate, although in the context of therapies, the monoclonal antibody that are neutralizing seems to have a better fate or advance more. So it's a very complex question, but I think ultimately if you have a good animal model that was just mentioned, which is very, very important, the most critical part, and you have a vaccine that do work, even if you don't know why, I think and many vaccines are likely to have different mechanism of protection as well. So again, like it's maybe I'm totally wrong on this one, but I would personally not focus, I would be nervous about focusing something specifically on broadly neutralizing antibody if I don't know 200% sure that this is really what is needed. I think that for Ebola or HIV, you actually need everything you got to beat it. and you want it all. You want the antibody, the T cells, the innate immune response, everything you can to get it out.
So this question for Jamie. It's me. So you mentioned B cell maturation in your experiment. So what do you think your DNA prime protein boost will help B cell maturation, and what might be the mechanism?
So you're asking whether or not my DNA prime protein boost protocol will help B cell affinity maturation. Well, I think the data suggests it does help B cell affinity maturation. I mean, we're doing it by looking at the avidity of the antibody. So that is it's a measure of the binding strength of an antibody to an antigen, which indicates we're getting B cell affinity maturation, but I can't say for sure that it's actually happening, but it looks like it is happening. It looks promising. We weren't expecting to see that. In fact, we didn't actually start looking at the avidity of the antibody responses until we started seeing really no differences between the responses, and then we were actually quite surprised to see that. So yeah, I'm quite optimistic that it would do.
So you it's not for my edification because I was there, but just to make clarification for the rest of the audience, what's the timeline right now to look at potential RFA or for a call for related vaccine research in HIV based on the meeting we have? And maybe you can explain the process a bit.
It's going to be rapid. So we do have money allocated for HIV vaccine, and now I'm trying to make eye contact with Michelle. Yeah, and this is what? Can you speak louder?
I would say the fall.
The
for a
fall.
launch is a good timeline to think about.
Like in the next 6 months or so. So this is, I mean, and we are in discussions. And we have some, I think, reasonable ideas that the workshop we held in Ottawa was very helpful. And I'm quite confident that what we will deliver will be of interest to the community.
So if there's no more questions, I guess I'll wrap things up. I want to thank all the speakers. It was some great presentations. Great to have them here and present their perspectives. I do have gifts. They're courtesy of the Alliance Coordinating Office. I think, Marc, you already received or you're in conflict if you can't receive gifts, right?
Well, actually, I took the liberty of having one.
You can't do it publicly. You can do it privately.